large value. And then the most contributor of the tuna fisheries is a tropical tuna, that where the two of them, skipjack tuna and yellowfin tuna, become our third and fifth ranks on the production by species in the globally. And in IOTC, the tropical tuna become IOTC become the second largest RFMO producer in the global tuna tropical tuna production. And Indonesia, in average, become the higher of the tuna production in the IOTC area. In the context of the FADs, fish aggregating devices, naturally, tuna and other some large pelagic species also aggregate the floating object. Based on this behavior, human-made FADs. FAD is artificial floating object designed to attract tuna and make them too easier to catch. And there is two FADs types in the world. It's drifting FADs and anchor FADs. And currently, around 38% global tuna production come from these fisheries. Also, around 90% operational operation in IOTC related to the FADs. And how about in Indonesia? FADs in Indonesia, also tuna fisheries in Indonesia. We only have anchor FADs in Indonesia, and we already started in 1980s for the tuna fisheries, and the development until now increased, and the massive development is started in 2004. At that time, we have a program on the tuna industrialization, and the deployment FAD is massive after that. Some issues that arise from the Indonesian tuna FAD fisheries. The fisheries are complex with the different fishing gear, various fishing gear, also the size, the scale, small scale until industrial, and the deployment is massive increase, making them too difficult to control, to monitor. And actually we have some several regulations related to the FADs, but the implementation is still challenges. And then we have national data collection, such as landing data and logbook data, but there is no information about the number and position of FADs. And because of that, I found the gaps. There's no study quantify the utilization of anchor FADs in Indonesia. And then I raised some scientific question: how much do the Indonesian tuna fisheries rely on the anchor FADs? The utilization of FADs, they have several impact beyond the positive impact, such as increased catchability is related to the economic revenue, fishing efficiency. They also have a negative impact, such as stock sustainability, because the purseiner and pull angler, they catch more juvenile tuna, so the fishing mortality of juvenile tuna more higher, and also increase the bycatch related to the shark, the vulnerability species. Also provide a degradation habitat, such as modification surface habitat and the marine pollution when they are damaged or lost. Related to the modification surface habitat, they also indirect impact of the physiological condition of tuna and also associated species that we call ecological trap hypothesis that already rise 25 years ago. Some explanation about the ecological trap hypothesis. As indicator log hypothesis, naturally tuna aggregate with the floating object like log, branch, algae, etc. from the river to the ocean that through some nutrient, and then log are indicator of high productivity or rich area. The massive deployment of the FADs modify the floating object component of tuna habitats, and then tuna will actively follow or stay around FADs, but they could attract or retain them in the less productive area that also can be unfavorable for their fitness. Some research that already provide previously related to the physiological impact of the FADs, the comparison between condition in FADs and out of FADs. First is from the stomach contents. What they found is tuna, when they caught in the FADs, is more higher empty stomach compared to the out of FADs. But it's still debatable because when they are caught tuna in the out of FADs, it's when they are fit. Also from the morphological and bioelectrical tissue, the condition tuna in FADs is lower compared to the out of FADs. But we don't know the lower is bad or good condition. It's already attained the bad condition or not. Also, recent study also mentioned as same things. The increasing the density of fats that maybe have impact on the condition of tuna, but we don't know the condition tuna is bad or not. Another study mentioned the increasing of the fats doesn't lead to the decreasing of in the long term, doesn't lead to the decreasing of the condition factor. The knowledge gap that I got it's not possible to assess whether the lower condition observed in fat associated tuna is good or bad condition due to the lack fundamental knowledge about the tuna physiology and dynamics. I raised two central questions from these gaps. How does the physiological condition tuna evolve in the time when they are not fit? Is representative of tuna in the fats condition, and what are the physiological characteristics tuna in good or bad condition? In the context of a tuna condition in the wild tuna, most of the study in physiological condition of tuna held in the natural floating objects, in the drifting fats, and in the Western Indian Ocean. They access several condition factors, including phase angle, KN, SSI, and the tissue composition specifically for lipid. But there is no study in anchor fats and in the Eastern Indian Ocean. So the scientific question that I raise is, how does the utilization of anchor fats in Indonesia affect the physiological condition of tuna? My general objective of my PhD study is to investigate the utilization of fats, specifically anchor fats, and their impacts on the physiological condition of tuna in the Indonesian fats array, anchor fats array. I divided to 4 chapters. The first chapter I characterize the Indonesian fats fisheries. Second, I did a physiological experiment for fasting experiment to know the physiological condition of tuna, and then I used the result from the fasting experiment to measure the physiological condition in the yellowfin tuna in the wild, and I test the condition factor in the wild. It is good or bad through a refeeding experiment. In chapter 1, it's related to the reliance and usage of anchor fats in the Indonesian tuna fisheries. I did some interview with the 293 fishers in several fishing ports, including small fishing port and industrial fishing port in West Java and West Sumatra and south of Java in Indonesia. And the target is fisheries that really relate to the fats. It's percent fishery, trolland, handland, longland, ex-longland fishery that already changed to the handland fishery. There is 4 main section of question. The first one is vessel identification related to the owner of vessel. And then the second is description of vessel, like a size of vessel, fishing practice related to the trip, how many trips, how long your trip, and then related to the anchor fats utilization, and then the last information, the last fishing trip. Based on our data, I try to group it in the 4 different groups with different characteristics. The first group is percent high seas. It's a percent operating in the EEZ, both EEZ and high seas. They have a more larger vessel, around 27, and the more larger fishing trip around 6 months. And the target is tuna. The second group is 4%, is percent EEZ, or the exclusively is EEZ. It's smaller vessel compared to percent high seas. Also, the duration is shorter, and the target, beside tuna, they also target small pelagic fisheries. The third group is more dynamic, hook and line, no like. They have a small vessel, also the duration fishing trip only 2 weeks until 1 month. They target, some of them they change the target with demersal fisheries, with the hairtail. Some of them they change the operational, become a support vessel for the purseiner. And the last group is hook and line using like. This is the group that previously using long line and they changed to be hand line. Some of them also, they changed the target, become hairtail and squid. I already published this this work on the regional studies in maritime last year. And what I found from the questionnaire is there is around 90% of the fishing operation of the fisheries is related to the anchor feds, especially for the Pursein, Hysis, and Pursein and who can land with no like. And who can land with like, usually they only few that they use feds because usually they use a like to attract the tuna and they drift on the out of feds. So I got information about the number of feds that they have per vessel. The percentage is around 8 feds, and then the others is less than 4 feds, 2 until 4 feds per vessel. And then all of the feds is privately. The owner of feds is a vessel owner, the company. Also, some of them belong to the group or association, some of them belong to captain, and they share the utilization of feds in the group or on the all of vessel. And when they share, some of them is just they have to share the cat, some of them just a permission. And I got also a result about the lifespan of feds. Anchor feds is around 2 until 4 years. It's represent they have a good maintenance for this feds. And so just for summary, so take home message. Indonesian tuna fisheries is very complex, but using the material, I think we can simplify the fisheries. And then the second is Pursein and Highland really rely on feds. So if we have like a ban or restriction of the feds, maybe will impact it to the fishing industry, to the fishing community, to livelihood of fisheries. And the fat is privately owned. They provide a good maintenance, so it will be have a long lifespan of fats, and then will reduce the marine pollution. But the worst is and they didn't want to share their cats, their data to us because they will want to keep because belong to them. My second chapter is related to the physical condition of yellowfin tuna, subject to food deprivation, fasting experiment. The experiment I conducted in October, December 2023. Within the 33 individual we measure, and the fog length is 26 under 71. The location in the experiment location in Gondol Mariculture Laboratory in North Bali in Indonesia. This is one of the two, I think the two captive tuna for tropical tuna beside the Panama. And how to get the fish? We catch fish from feds in north of Bali, and we bring them to the tanks. We divide to two tanks. With the size of tank is the diameter is 8 meters and the depth is 3 meters. And then we maintain them, we captive them, we feed them at least for 2 weeks, and then provide the fasting for 5 weeks, and we measure them 3 until 7 individual at regular 3 until 6 days. What we measure? We measure several physiological conditions, biomarks, just to know which biomarks can really identify the movement, the development of the physiological condition. And then that really gives us a signal the condition is changed. From the live tuna, we measure several condition factors such as we measure the swimming speed from the video analysis. We also measure the pitch angle using bioimpedance analysis, and then we measure several blood composition. When the tuna die, we also measure the Lagrange condition factor is related to the weight compared to the weight, theoretical weight. Hepatosomatic index is liver weight compared to the weight of fish, and then gut length ratio. So we took a tissue sample, and we analyzed the proximal analysis to know the percentage of lipid, protein, or water content. First, what we got is about the rated the swimming speed. I use EA to identify the tuna and then to identify the movement, and then I calculate the median speed in the 1 minute regulating interval. I only take one sample during 1 hour. in 1 minute. And then what I got is there was a significant difference in median swim speed between the pre-fasting during captivity and a fasting period. When I look at during the fasting period, there is decreasing trend in the beginning gradually and stabilize around day 4 until the end of the experiment. I only use the data from the diet fish. I try to analyze using a multivariate analysis with 83% variant explanation variance. And then from this analysis, I can characterize, I can provide the group of the condition. We divided the 3 groups. It's related to also to the physiological of fasting basis in the several animals. And the group 1 is representative. The group 1 is more correlated with the positive correlated with the tissue lipid, tissue protein, gut length, and Larkin condition factor. And group 2 is more negative correlation with the SSI. And group 3, I only found 3 individuals that in attaining in group 3 related to the positive correlation with the tissue waters. Based on this group, I try to look at another physiological condition, such as a morphometric. For the morphometric, I analyze the hypotosmotic index, Lagrange condition factor, and gut length index. All of them is they are significant decrease during the fasting experiment. And then the analysis also revealed that distinct temporal phases in the condition factor. In the first is the decreasing on the SSI in the day 4, around day 4, and then decreasing the KN and gut length more longer in day 10 until day 16, and then after day 16, the SSI and KN also gut length is low. There was a significant difference between in SSI, KN, and gut length between group 1 and group 2, and there is no effect of the fish size on this result. For the tissue condition, the base angle is decreased more late compared to the morphometric. It's around day 25. The lipid is decreased around after day 4 also, and the protein is decreased also significantly, and the tissue water is increased. And there is no significant difference in base angle between group 1 and group 2. And there was, but in the others, there was a significant difference. And to be considered, there was some effect on the fish size, on lipid and tissue water, and water tissue content. From blood biomarkers that we measure, we have several biomarkers, blood biomarkers, and I try to analyze using a multivariate analysis with around 47 explanation variants for all blood parameters. And there is some bloods that correlated with the PC1, such as protein, cortisol, hematocrit, lactate, 3G, cholesterol, and glucose. And then in the other set is glucagon, and then FFA urea ketone is more close to the PC2. From this result, I also tried to provide the trajectory analysis. And in the first 13 days, mostly they are fluctuated mostly in the right side with the PC1, and then after that, after 30 days, they move to the left, most in the shift to the negative on the PC1, accompanied by in the decrease in hematocrit and protein and increase in the glucagon. For summary of my result in the fasting experiment, from the group, we can define also the pace that we can characterize the pace of the development of the physiological condition of tuna. And there is 3 distinct phases of the energy utilization. The first is the tuna rely on the energy source from the liver. It's characterized by decreasing the liver weight, the SSI, and that time also the speed also decreased. And the second is the second phase is the utilization fatty acid stored in the adipose tissue. It's characterized by decreasing KN and GERD length. Also, the lipid content from the tissue is decreased, and at that time they change some blood parameters. And then the last phase, the phase 3, tuna begin mobilize the muscle tissue characterized by decreasing the phase angle, and then all of the parameters become low. For my third chapter, it's related to the physiological condition of wild yellowfin tuna in the Indonesian fats array. Right now, we know from the experiment, we have a several condition factor. We have a different basis of the condition factor. And I just try to look at the two condition factors that really easy to measure in the wild. And I collect some data in the wild in Aceh, in West Sumatra, during April 2024, around 128 fish. And then another location, I collected data in Pancar, south of Java, in June, October 2025, around 145 fish. And from 2 dimensions of SSI and KN, I try to plot it, and then we can see the phase 1 is the movement of the condition factor between phase. Phase 1 is in the high SSI and high KN, and then phase 2 move to the low SSI but the KN still high, and then phase 3 is a low in the both KN and SSI. When I overlay data from the wild tuna, what we got is it's around 90%. They are still in the phase 1, and only less than 10% they're training in phase 2. I try also to compare data from IOTC from the Western Indian Ocean. And what I got is around 78% still in the phase 1, and only 32% attaining the phase 2. And what I summary from this work is most tuna around fights have a similar condition to captive tuna, subject to less than 4 days of fasting. It means in the phase 1. And then there is no tuna in the low SSI and low KN. And two hypotheses that I raise is tuna leave the fats before attaining the lower condition. It means we not really support the ecological trap hypothesis. Or the worst case is tuna die before attaining the lower condition is related to the higher natural mortality in phase 2 or phase 3. For my last chapter, so it's related to the visual recovery of yellowfin tuna through a refeeding experiment. So right now we got the condition in the wild that tuna mostly in the phase 1 and only 10 until 20% tuna in the phase 2. And we know the characteristic of the phase 2 during a fasting experiment. We know some biomarks, some condition factor, but we don't know if this in this phase is a good or bad condition. So I tried to provide the experiment, refeeding experiment, to know how fast they recover. The experiment conducted in January and until March 2025. There's 33 individual measure with fork length more higher, more larger, 34 until 64 centimeters. In the same place, also to collect the sample is the same methodology to maintain them steam, but the experiment is different. We fasting them for 7 days and we refeeding them at day 8. We measure 3 until 4 individuals measure after 7 days, and after day 7 at regular interval 1 until 3 is more shorter to really know the development of the condition. What we measure is the same with the fasting experiment. What we got is for the swim speed, the median swim speed of yellowfin tuna increased after being fed again. It's more active again after we feed them, and then from the morphometric, the declining of SSI serve as that's the earliest signal from the fasting experiment that going down. They increase quickly upon the refeeding. And another condition is KN value is from the refeeding remain within the same range between the refeeding and the beginning of the fasting treatment. And we will compare the refeeding with the base one is there is no significant difference. So based on that knowledge, the summary is yellowfin tuna associated with fats are still in good condition, demonstrated by the rapid recovery once they resume feeding. But yeah, we still need additional experiment for the next extended duration of the fasting in phase 2, might be in day 14 or until day 20, or maybe in phase 3 to know the really condition, it is good on or bad condition. For the conclusion of my PhD project is the first is Indonesian tuna fisheries are highly dependent on the utilization of fats, so we have to maintain this fisheries. However, we need to improve the data collection program. The second is the fasting experiment have a provide clear insight into the development of the physiological indicator of yellowfin tuna. We can use this indicator to measure condition factor in the wild. And then the third is the use of anchor fats doesn't appear in the negative impact on the physiological condition of yellowfin tuna, as individual around fats remain in good condition and show rapid recovery when the refeeding resume. But we must also consider other impact of fats, such as sustainability, the juvenile, the bycatch, and the habitat degradation. So we still have a need fats management. That's all my presentation. But before I close, I just want to show you some video in the internet. It's related to the how sensitive to now with the manipulation with the environmental. So when they have a like, to not more stressed and they hit the wall. It's also happened in our experiment. That's thanks all. Thanks for my supervisor, our team in here, also in Indonesia, in Gondol, Bali, and thanks for Manfaat Project for funding. Thank you. Thank you very much.
Thank you very much, Matthew. Some questions? Okay.
Many thanks. Congratulations for this very nice work. I just have a question. You did compare with tuna coat in unsure fats. That do you have samples out from this fats? I mean that it will be very interesting to see how in which types they belong when they are coat out from the fats.
Sample from Indonesia we don't have because mostly we use the fats and then there is no like a research in the out of fats that we found. But I think in IOTC they have a sample in pre-schooling sample, but I didn't use that one to compare. I think just in this IOTC. But I don't have in Indonesia, so I did.
Thank you for your presentation. I have a question about anchored fat in Indonesia because I don't know exactly how it works. You said it was mainly privately owned.
Yes.
And I was wondering how can they know that it's only used by them? I mean, do they have a kind of surveillance that it's used by them, or they just say it's them, but they don't know if artisanal fishers are using it?
What I found also in I already published in my first chapter is they have a collaboration. The IRL said before they have collaboration between purseiner and handlander. Usually, yeah, they don't know who will use their fats, but usually they collaborate. They ask to handlander, decide they catch the tuna to like a service to the purseiner to guide, to like protect their fights, to inform their fight if there is some fish in the fights, to contact the purseiner to come, and then the purseiner come and sell to the headliners. So they have collaboration. Also, they have a collaboration between the group. So they change the group, like they have different days to attending the fights, and then just to like protect their fights. But sometimes they also, we don't know in the sea. Sometimes they also catch the other fights.
But how far it is from the coast?
Closest is I It is around 10 nautical miles. In the western is more far. For the percent high seas is more far. It's the high seas, but for the percent EZ and Hook and Line, yeah, the closest is around 10 nautical miles, I think. No, 30 nautical miles from the coast. Yeah.
Thank you.
Yes.
This is a really cool thesis. I really liked it. So I'm interested in, you said your fads last 2 to 4 years, which is longer than I've heard reported elsewhere.
Yeah.
I'm wondering if you have looked at all what happens at the end. Like, why does the fad stop being maintained or break, or what is the final event? And I'm just curious, is there any centralized database that the Indonesian government has or somebody has of where all these fads are or how many there are?
Yeah, the first question for the maintenance, they actually every single, like not really single, but when they go to the sea, they always provide the reunion, the tractor, the rope. They always check and then they always repair them before the cause of the loss of fats is the wave, the current. If they have a big current, also when the longliner cut the fats, that's mostly impacted to the loss of fats. And for the database, right now we don't have really data about the fats installation, fats where they deploy, where they how many fats we deploy. We don't have any information, even though only the number of the fats, because we have a regulation regarding the report fats. We have to report to the registration to register to the government, but the officers is like he doesn't want to register something, so it's difficult to us to collect the data. So yeah.
Questions?
Thanks, Igna. I also would like to thank you because I remember the beginning of all these experiments, and it's really nice to see.
I saw your picture here.
Oh, it's okay, that's all right. So I really like the story that you can tell now. But I had one question regarding the physiological condition of the tuna in the wild. The two results that you showed are actually snapshots that you measure at
the same time.
At the same time. Yeah. But I was wondering whether you had the opportunity or any idea of whether this condition could vary in a different season or along the year. Do you have any other samples besides these two periods?
Yeah, right now we start to looking for funding to collect another data. We will test. There is in the North Bali, there is a gradation of the fats density from the western to the eastern, and we try to analyze that there is any impact of the density of fats on the physiological condition, and we will measure more longer time, like maybe 1 year in the north of Bali. So maybe that will be answer the question. But right now we don't have a we don't have the key end, but we don't have mostly what data that we collected is only related to the key end, but the SSI, the liver weight, we didn't collect. So yeah, we will do it if we got the funding for the next research.
Questions? Maybe online? Some questions? No. I have one.
Yes.
Because you said that the fisheries were relying a lot on fats, and you made some interviews. Did some fisheries express some concerns about that? About, for example, the local knowledge of fisheries that was being lost because of that, strong reliance?
Yeah, they really strong reliance. Yeah, they don't want to they have no fats in the at all. You mean like that, or what you? Yes.
Are they concerned that
But
the fishers that they are too reliant on the fats and
Yeah, the concern they rely on fats. And actually, they want to when we discuss also the research, they want to register the fats, but the difficulties right now is in the process of the registration, fat registration. And yeah, right now we try to we have several discussions with the ministerial to provide a more easier to register. So they really know that they really depend on the fats when they are fishing when they fish a tuna. So yeah, I don't know. It's answer your question.
well, it was more about are they preoccupied because of that or not? Like, do they worry that there are too?
Do they worry about
If the stock crash, for example, what if the stock crashes, for example?
Do
Do you have any
different?
alternative way of fishing or any other species or?
For the small scale, yes, they sometimes they chase with the target species, but I think for the industrial, no, they still want to do it.
The questions? No. Maybe online? Some questions online? Yes. Classical. Okay. Well, thank you very much.
Thank you very much.
Thank you for coming.
À l'aise.